Look at the beautiful white flowers of this begonia wax. Just fabulous. Okay, now that we're all happy, I'm going to talk about my project, my leaf cutting project that was made on Peperomia Argyria. So that's a Peperomia Argyria. You can see the leaves are cupped a bit and ovate. The leaves are dark striped green with silvery center. Since there are more than 1,000 species of Peperomia, they all comes with different characteristics, but most of them are evergreen and perennials. Since they come from the tropics, they really like higher temperature, like around 20 degrees, but they're really sensitive to water saturation since it inhibits their oxygen and increase susceptibility to fungi. So they're mostly used for indoor ornamental, as indoor ornamental plants. Like we can see in the video, the branch are mostly alternate. So our objective for here consisted to see how the leaf cutting size affects the propagation success rate. So for example, let's say our grower only have one stock plant having 60 leaves. Will it be possible to cut the leaves in two in order to have like 120 new plants? So here are three treatments that will be tested. Will consist of an entire leaf, the top portion of the leaf, and the bottom of it. The trial started on January the 27th. The substrate was a combination of moist G6 and perlite soil. Before I started the experiment, the knife, the tray, and the workplace got sterilized. The leaf blades was kept while the petiole was left on the mother plant. Careful manipulation was required to avoid any damage to the leaf blades. Further cutting were required to obtain my top and bottom leaf portion. The usage of stem root number 1 was used since it promotes the root formation according to the literature. My experimental design consists of 3 replicates for my 3 treatments, while 10 leaves were put in each row. The tray was put on the open bench with a plastic cover on it in order to increase humidity. The cover was opened in the following weeks in order to allow air exchange. The workplace got clean, the job was well done, everybody's happy, and monitoring could start. No! Leaf necrosis occurred only 1 week after the beginning of the experiment. It was probably due to the presence of a fungi since white spore appeared on the surface of the leaf cuttings. The leaf became even worse as the week went by, and there was any formation of roots after 17 days. So my beloved cutting ending up at the bottom of a trash can. So on February the 13th, the same experiment was made again. The tools got sanitized another time, but since there was not enough Peperomia Argyria leaf cuttings, one replicate of Peperomia obtusifolia, which is the plant on the photo, replaced the original leaf cuttings. These leaf cuttings of the Peperomia obtusifolia have more of a round shape with a dark green color. On this time, plastic row cover was not used, and I was most careful in picking up healthy leaf cuttings. The evaluation was made on March the 24th. The leaves were carefully removed from the substrate with the roots and laid out on a table for further evaluation. Surprisingly, the leaf cuttings of the obtusifolia, being the darker leaves, seems to be in good shape, but most of them did not form any roots. On the other sides, the leaf cutting of the Peperomia Argyria seems to be really in bad shapes, but most of them had roots, like we can see on the picture. Again, the objective of the experiment was to see how the size and the location of leaf cuttings affect their propagation. The percentage success rate of propagation, biomass grading, and maximum root length were evaluated. As we can see on the graph, there was 3 different colors that differentiate the different replicate in the tray. On the first graph, we can see that the top portion of the Peperomia leaves were the most successful in making roots, while 75% of the leaf cutting had roots. On the other side, the bottom and whole part of the leaf have almost the same propagation rate, but being lower than the top leaf portion. For the biomass, on the next graph, it's the biomass grading, where leaf cutting of every row were evaluated on the scale from 0 to 3. 0, there was no roots whatsoever, while 3, there was a complex root system associated with the leaf cutting. So here again, the top leaf portion had the best biomass, followed by a whole leaf cutting, and finally the bottom leaf portion was the worst in terms of biomass. And the third graph, it's representing the maximum length of the root from each leaf cuttings. The ranking was the same than in the second graph. So first is the top leaf portion, the second one is the entire leaf portion or whole leaf cutting, and then at the end it's the bottom leaf cutting. So here, what's the conclusion that we can take home is that grower can successfully propagate fewer initial leaves by cutting them in two. So although the bottom portion of the leaf struggled a little bit more, the top portion of the leaf had an even higher propagation rate than propagating a whole leaf. So it's a process that can be made. I would like to thank Daniel Donnelly for her time and patience. Thank you.